Welcome friends and welcome to my channel Sequential Biology. We are talking about bacteriophage. In the previous two videos we have discussed about structure of bacteriophage and also different types of bacteriophage. And today we will be talking about the life cycle pattern of bacteriophage. Actually bacteriophage follow the two types of life cycle pattern. One is lytic cycle and another is lysogenic cycle. And here we will discuss about lytic and lysogenic cycle. First we will be talking about the lytic cycle of bacteriophage. The lytic means lysis. That means when any bacteriophage attack a bacteria, this will causes a rupture of this bacterial cell. Suppose this is a bacteria, and when these bacteria are infected by bacteriophage, this is bacteriophage, and this bacteriophage penetrate their genetic material inside their host bacteria, and then they replicate inside this host cell, and after formation of many progeny viruses, and they rupture or kill this bacterial cell or the host cell. And the process of life cycle pattern by which any bacteriophage kill their host cell, this process of this life cycle pattern is known as lytic cycle. And this lytic cycle is performed by 5 steps. The first step is attachment. That means the bacteriophage must be attached with their host cell. But how they attach with their host cell? Attachment of the bacteriophage is also known as the adsorption and the receptor of the host cell wall. The host cell have specific receptor, and with the help of this receptor, the bacteriophage attached with this host cell. And after then, the lysozyme is a kind of enzyme secret from the ends of the phage tail fiber. Okay, the lysozyme is a type of protein which destroy the bacterial cell wall, and by destroying this bacterial cell wall, they penetrate their genetic material inside host cell. Then this lysozyme are secreted from phage tail fiber. At the end plate or the basal plate, there are present many tail fiber, and this tail fiber actually secrete this lysozyme, and this lysozyme causes a breakdown of this bacterial cell wall. Cell wall of bacteria is made up of peptidoglycan. Peptidoglycan is a special type of polysaccharide, heteropolysaccharide, and this lysozyme break this peptidoglycan structure. Okay, and the second step, then this is the first step, is attachment, and bacteriophage attached with their host cell by their specific receptor. Second is penetration or injection. Then this is DNA. This is virion. Virion means a complete infective particle of virus is known as virion. And in this virion, you can see the DNA is present just in the bacteriophage, and in the next step, the bacteriophage penetrate their DNA inside host cell, and this process is known as penetration or injection. Then this penetration or injection is occurred by contractile sheath membrane. The infective virion before penetration is called extracellular virion. Then before penetrating this virion is called as extracellular virion, and the bacteriophage genome enters cytoplasm of host cell. This is bacterial genome, that means the viral genome, bacteriophage genome. So this bacteriophage genome enters into host cell, and this is known as penetration or injection. Synthesis of virus, nucleic acid and protein. When the bacteriophage penetrate their genetic material inside host cell, this hijack the host cell protein synthesis machinery, and after then they synthesize their own protein, their own viral protein. The bacterial protein will not be synthesized, the viral protein will be synthesized, and this is capsid, and also this DNA will be replicated inside the host cell. So many copy of this progeny DNA. These are the progeny DNA, and this progeny DNA will be synthesized inside host cell. So synthesis of virus, nucleic acid and the protein. As the genome enters into host cell cytoplasm, it subverts the host nucleic acid. So this will inactivate the host nucleic acid and also the protein synthesis machinery apparatus, and initiate synthesis of viral DNA and viral protein, right? And next step is assembly and packaging. Then this is second step, this is second step, and this is the third step. And after formation of this different type of protein or the capsid and the DNA, and this DNA and capsid will be packaged, and this form a complete virus particle. As the virus protein are synthesized, they self-assemble into the viral components such as the head with DNA, tail and the tail fiber. So different structure will be formed, and this is known as assembly and the packaging. And after then, when this bacteria will be will be full up with the bacteriophage. Then this bacteriophage or the virus rupture this bacterial cell, and this is called as release or lysis. The average number of phage particle produced by each infected cell, and this is known as burst size. Whenever these bacteria will be loaded by 50 to several hundred bacteriophage, then the cell will be ruptured and the viral protein cause the lysis of the host and release into the environment. Then these are the different steps, these are the different process which are followed during lytic cycle. The first one is attachment, second is penetration or injection, and this is the third step. Third step is synthesis of virus, nucleic acid and protein, and this is fourth step. Fourth step is assembly and packaging, and the fifth step is the release or lysis. Okay, and this is the process by which bacteriophage perform their lytic cycle. And the another life cycle pattern is the lysogenic process. In lysogenic process, when we have seen that in lytic cycle, the bacteriophage DNA hijack the protein synthesis machinery of the host cell, and later this will rupture the host cell. But in lysogenic cycle, the bacteriophage DNA or genome will be replicated with the host genome. This will not destroy the host genome. The bacteriophage or the T4 is virulent, and some other viruses, although able to kill the cells through the lytic cycle, can also undergo different life cycle pattern, resulting in a stable genetic relationship with the host. And such viruses are called as temperate phage. So the phages which show lysogenic cycle in their life cycle pattern are known as temperate phage. But the phages which show lytic cycle are known as virulent phage. But the phage which show lysogenic pattern of their life cycle, they are known as temperate viruses or temperate phage. Such viruses can enter into a state called the lysogeny. Okay, so this process is known as lysogeny. How this lysogeny is performed? This is a temperate phage, and in this temperate phage you can see this is capsid. So this is capsid or the head, and inside head their genetic material is present. Their genetic material or the DNA is present, and then they penetrate their genetic material inside host cell. Then this is host cell DNA, and this is viral DNA, or this is bacteriophage DNA. Then this DNA enters into the host cell, and in lysogenic cycle, after entering into host cell, this DNA will be incorporated into bacterial DNA, and this special structure is known as prophage. Okay, so when the bacteriophage DNA incorporated into bacterial DNA, and this will form a special genome, and this is known as prophage, and the viral DNA is integrated into host DNA, and this will form the prophage, and the bacteria which contain a prophage is known as lysogenized cell, right? The bacteria which contain this prophage, that means in their genetic material you can find the viral DNA, and these are known as lysogenized phage. And when this bacteria undergoes their reproduction or the cell division, then the viral DNA will also be replicated with the host genome. Okay, and this is the process by which they increase the number of their genetic material. And sometimes this viral genome, sometimes this viral DNA will be released from this prophage. So this viral DNA will be released from this prophage, and this will replicate inside host cell and also hijack the protein synthesis machinery, and thus they will produce many coat protein and also other structural protein inside their host cell. Consequences of infection by temperate bacteriophage and the lysogenized cell can be induced to produce the mature virus and lysis. In lysogeny, most virus genome are not expressed. In lysogeny, most virus genome are not expressed in host cell, and the virus genome are called as the prophage. Then the virus genome are known as prophage, which is inserted into host DNA and reproduces as a host DNA replicate. When the host DNA replicate, this will replicate with this host DNA. A lysogenic virus or temperate phage can remain in this state for numerous replication. Okay, for numerous replication, they remains in prophage stage of the host cell and of the host cell DNA until it excises. Excises means the release of viral DNA from this prophage and itself from the host DNA and undergoes the lytic cycle. When the genetic material of this phage is inserted into DNA of the host cell, it is said to be prophage stage, and a cell that contains the prophage is known as lysogen. This is very, very important. What is prophage state and what is lysogen? The physical state of the prophage is not identical for all temperate phage. The prophage of the lambda in E. coli is integrated into bacterial chromosome at specific site, whereas the prophage of P1 in E. coli replicate as an extra chromosomal plasmid, right? What is this plasmid? Plasmid are nothing but this is an extra chromosomal DNA inside host cell or extra chromosomal DNA inside bacteria, and usually this plasmid contains many antibiotic resistant gene, right? And this will protect this bacteria from antibiotic. And when the viral DNA enters into this bacteria, and sometimes they replicate like extra chromosomal plasmid. The plasmid is a self-replicating structure, self-replicating DNA. In lysogeny, the phage genome is replicated along with host genome during cell division, is passed from the next generation. The importance of this lysogeny: most bacteria isolated in nature are lysogen. That means they contain viral DNA for one or more bacteriophage, and this will causes genetic recombination. Lysogeny may confer new genetic property of bacteria since viral DNA incorporated into the bacterial DNA. So a new genetic recombination can be seen in case of bacteria, and for this reason the lysogeny may confer new genetic property to the bacteria. That means they actually diverse this bacteria. Okay, they diverse this bacteria and they produce different strain of bacteria. In pathogenic bacteria, the virulency maybe depends on lysogenic bacteriophage as they harbor. Now we will discuss about process of lysogeny or the genetics of lysogeny. Bacteriophage lambda infect E. coli. The lambda genome consisting of linear double-stranded DNA. This is a linear double-stranded DNA. And at the 5' end of each strand is a single-stranded region of 20 nucleotide long, and this single-stranded cohesive end are complementary. And when the lambda DNA enters into host cell, they base pair and forming a cos site, and the DNA is then ligated and forming a double-stranded circular. It's a linear DNA and which is injected into the host cell, and then this linear DNA will be circularized and then integrated into the host DNA, and this will form prophage. Okay, actually it has this linear DNA have two cohesive end, and since there are presents a cohesive end or the sticky end, so easily it will be circularized, right? The integration of the circular lambda genome involves the presence of attachment site, but how this circular lambda genome incorporate into host genome, this is very, very important, and we will see the genetics behind it. The integration of circular lambda genome involves the presence of attachment site. Attachment site in the genome of both bacteriophage and the bacteria. Suppose this is an bacteriophage DNA, so this is an bacteriophage DNA and this is bacterial DNA, and each of this DNA have attachment site. This is phage DNA attachment site and this is bacterial DNA attachment site, and in both these attachment site the special type of structure are found. In bacteriophage we can see POP structure, and in bacterial DNA or the host DNA BOB structure. These O are common. Okay, and with this attachment site, actually the phage DNA will be incorporated with this attachment site. The bacterial attachment site is called as attachment B, and consisting of specific DNA sequences consisting of 3 domain BOB dash. Then the bacterial DNA or the host DNA contains attachment B site, and this has 3 important domain or the 3 important sequence BOB dash. The phage attachment site is attachment phage and is also made up of 3 domain POP dash domain. The O domain is common to both phage and the bacteria and called core sequence. So this O sequence is known as core sequence. Now the lysogeny or the lytic phage due to the race of 2 protein. When the lambda repressor protein prevails, lambda repressor protein are synthesized from the CI gene, and when this lambda repressor protein prevails, this promote lysogeny, and when CRO protein prevails, this promote lytic cycle. When this lambda repressor protein prevail, that means when the lambda repressor protein is more lambda repressor protein is more than CRO protein, then we can see lysogeny. Then bacteriophage choose the lysogenic cycle. And when this CRO protein is more than the CI protein or the lambda repressor protein, then this bacteriophage again enters into the lytic cycle. Then choice of lytic and lysogeny actually is occurred due to race of two protein, the lambda repressor protein and CRO protein. When lambda repressor protein prevails, then this will promote lysogeny, and when CRO protein prevails, this will promote lytic cycle. So this is all about life cycle pattern of bacteriophage, and in the next video we will discuss about COVID-19 pathophysiology. COVID-19 means coronavirus infected disease 2019. Thank you, and that's it.